Hi, everyone. Welcome to Biopsychology. We're going to be talking about Chapter 4. We'll do the first half of Chapter 4 in this video for this week, and I'll post the second half toward the end of the week. But we're going to talk about neural conduction and synaptic transmission. So, how neurons are sending signals, how they're receiving signals. And you and I, all of us, we communicate through spoken words. When it comes to neurons, they are actually communicating through chemicals, through electrical signals. And so let's start diving into what that looks like. Now, I want to talk about the idea of a membrane potential. And that might sound really intimidating, but think of it like a charge on a battery. So I'll try and use this analogy throughout. But every cell that we have in our body, especially neurons, has a tiny difference in electrical charge across this membrane. Okay. And that difference is called the resting membrane potential. So, what exactly is it? It's really just the balance of positive and negative charges that are inside the cell compared to the outside of the cell. And when we're at rest, or when the neurons are at rest, the inside of a neuron is actually more negative than the outside. So, just like a battery storing up its energy. Now, how do we measure it? Scientists use very fine electrodes. They're almost like tiny little probes to measure the difference in voltage. And you don't, need to remember the exact tools here, but the key idea is yes, we can actually measure this voltage difference or this electrical energy in real time. Why is it important? Because without this charged battery, your neurons couldn't fire. So, that resting membrane potential is the foundation that allows nerve cells to send signals, muscles to contract, and even, heart to beat. Without it, the communication system in our body will actually shut down. So, if you think of neurons like text messaging, right, the resting membrane potential is what keeps the phone charged. Without a charged battery, no messages are going to get through. So we just said resting membrane potential is like the cell's battery, but what actually creates and maintains that charge? So to answer that, we need to look at what's called the ionic basis. And basically that is how ions like sodium and potassium, so sodium Na plus and potassium K plus, move in and out of the cell. Now, think of sodium and potassium as two types of players. They're on opposite teams. Sodium is hanging out more on the outside of the cell, and potassium is hanging out more inside the cell. Now, this uneven distribution actually sets the stage for the electrical charge difference. What is diffusion pressure? So let's talk about that for a quick sec. But this is nature's way of evening things out. So just like if you are in a room and you spray perfume in one corner of the room, the scent spreads everywhere. And so sodium and potassium want to move from crowded areas to less crowded ones. The electrostatic pressure is really just about charge. Opposites attract, likes repel. So positive ions like sodium, potassium, they're influenced by the fact that the inside of a cell is more negative. And that negative inside is attracting them in, like a magnet that's pulling them closer. Now, the cell membrane isn't just a solid wall. It actually has special doors called ion channels. And these doors let ions move in and or out, but really only at the right times. So you can think of them like a revolving door at a hotel lobby, right? They're controlling the flow of what's coming in and out. And then we have the sodium potassium pump, and this is the hardest worker of all. It's like, think of it as a bouncer in a club, right? They're constantly pushing, sodium out and pulling potassium in. They're using energy to keep the balance just right. And without this pump, that battery that I mentioned would drain, and then the neurons couldn't function. So here's a picture of what is really going on with sodium and potassium around the neuron. And there are three main forces at play here, important to note. There is the diffusion pressure, so concentration gradient. So if you look at the sodium ions, which are Na, those are outside, and you look at the potassium ions inside. Okay. Now, nature always wants its balance. I just mentioned this. So, sodium is trying to get in, potassium is trying to get out. That is that diffusion pressure. So, just like how that perfume spreads across the room. there is the electrostatic pressure. And again, right, the inside of the neuron is negatively charged compared to the outside. Okay, so positive ions like sodium and potassium are attracted to that negativity, just like magnets. As I mentioned, this is what it looks like in the figure. Okay, and then I talked about the sodium potassium pump, right? So the hardest worker of all. It's working constantly to move those ions in and out. It's using energy. Okay, one thing to note is that for every 3 sodium ions it kicks out, it brings 2 potassium ions in. And this pump keeps the imbalance going on purpose. And that imbalance is exactly what creates the resting membrane potential, right? So that cell's little stored battery. So if you think about it, the concentration gradient is saying spread things out, right? Spread out, balance things. The electrostatic pressure is saying, hey, positives, come inside to the negative. And the pump is saying, nope, I am going to keep sodium out and I'm going to keep potassium in because that's how the neuron stays ready to fire. And that tension between all three is what maintains the resting potential. So we've seen how the neuron sets up its battery with sodium, with potassium. Now let's move to the next step. So what happens after the battery is actually charged? This is where communication between neurons actually happens. And that's through something called the postsynaptic potentials. Now, postsynaptic potentials is when one neuron wants to send a message to another, it actually releases neurotransmitters into the synapse. And that is the little gap between cells. So if you remember from one of our first chapters, I talked about how the dendrites, those tree-like branches, are actually bringing signals in to the cell body. A decision is being made in milliseconds by the axon hillock. It is going through this kind of like transmission cable super, super fast, and then it's heading down towards those buttons or those axon terminals. There is a space, I mentioned a couple, maybe last week, that 2 neurons don't ever really touch, but there's a synapse, right? And that gap is there. So that is the gap between the cells. Now, those neurotransmitters, what's happening is it's actually binding to receptors on the postsynaptic neuron. So if you think of that is the I guess neuron that is receiving the signal or that fire from the prior neuron. But that creates a tiny electrical charge, and that's what is the postsynaptic potential. So basically, it is receiving the message from the neuron saying, okay, I got your message, I'm ready. Now, there are a couple things I want to go through because there are 2 types of potentials. There is excitatory postsynaptic potentials, and it's also known as ES or EPSPs. And an EPSP is like pressing on the gas pedal. It makes the receiving neuron more likely to fire its own signal by bringing it closer to that threshold that's needed for the action potential, which the action potential is, are we good to go, right? Is that threshold there for us to actually fire? And then we have the inhibitory postsynaptic potentials, and those are IPSPs. On the other hand, that is like pressing the brake. Okay, or you can think of an EPSP as the green light and the IPSP as the red light. Now, it's making the neuron less likely to fire by pushing it further from that threshold where it could fire. Okay, so every neuron is constantly balancing gas pedals and brakes from all the signals that it's receiving. There are also 3 important properties that I want to talk about. So there are graded, decremental, and then transmission is rapid. And we'll talk about rapid. So graded. So postsynaptic potentials are not all or nothing like action potentials, right? They can be small, they can be large, depending on how much neurotransmitter is released. For the second one, decremental, they fade as they travel. So think of ripples in water that are getting smaller and farther out as they go. And then rapid. So transmission is rapid. And when I say that, it happens really, really fast. So neurons have to keep up with thousands of inputs every single second. So the process is lightning quick. So now that we've talked about the excitatory potentials, the inhibitory potentials, in theory, let's look at what they actually look like when we graph them. So this figure that you see here shows how the cell's membrane potential actually changes in response to stimulation. So again, we have to have something that happens that is actually stimulating this whole process. Now, to the top graph is going to be your excitatory postsynaptic potential. So here's an EPSP. And if you notice how the line goes up a little after the stimulus, right? After that stimulation, you see that go up. That means that the inside of the cell became slightly less negative. Okay, so it actually moved closer to the threshold for firing. That threshold for firing is going to be, I believe it's negative 55. So when we're at the resting potential, it is negative 70 millivolts. Okay. And so, um, again, the inside of the cell becomes slightly less negative, right? It's moved closer to that threshold for firing. So think of it as giving the neuron a gentle nudge forward, like pressing the gas or pedal. The middle graph is the inhibitory postsynaptic potential. So that is an IPSP. Now you're going to see the opposite happen. So as the line, you go through a stimulus, it's actually going down. It's dipping down after that stimulus. And that means that the inside of the cell became even more negative, right? So further away from the threshold. And this is like tapping the brakes, right? It's slowing the neuron down. It's making it less likely to fire. And then the bottom graph is an EPSP, so the excitatory, leading to an action potential. So when an EPSP is strong enough, or when several small EPSPs add up, the neuron reaches that threshold to fire. And that's when you get that big spike that you see on that bottom graph. That is what's called the action potential. This is an all or nothing signal. You can think of it as like there's no returning from this, right? We're firing off. This is the moment that the neuron actually fires and sends its message down the axon. So think of the neuron like a decision maker, right? The excitatory postsynaptic um, potentials are really the voices that are saying, yes, go ahead. The inhibitory ones are the voices that are saying, nope, we need to hold back. And only when the yes votes outweigh the no votes enough to reach the threshold does the neuron actually shout out its signal with that action potential. Again, it's important to note that at rest, it is negative 70 millivolts. Okay. So let's bring this together and talk about how postsynaptic potentials actually lead to that action potential. So action potentials always start at a special spot on the neuron cell, which is called the axon hillock. And I talked about this in class, and we'll see a figure in the next slide here. But that's basically that decision point where the neuron decides if it has enough input to fire. Okay. And remember that action potentials are all or nothing, right? Point of no return. The neuron doesn't kind of fire. It either fully fires or it doesn't at all. So just like flipping on a light switch, there's no halfway there. Let's talk a little bit about neural integration of multiple signals. This is where things get really interesting. A single EPSP on its own usually isn't enough to make a neuron fire. Neurons are constantly adding up inputs from thousands of synapses. And this process is called neural integration. Now, you have spatial summation, and this happens when signals from different places on the neuron arrive at the same time. So you can think of it like different friends that are all shouting go all at once, right? The combined effect of that is stronger. Then you have temporal summation, and this happens when signals from the same place really arrive one after another in quick succession. So imagine those friends. One friend is saying, go, go, go really, really fast. That repetition is also making that signal stronger. And location does matter, okay? So inputs that arrive closer to that axon hillock, which is that decision point, have more influence than signals that arrive far away from those dendrites, those tree-like branches. It's like being closer to the microphone, okay? Your voice carries more weight in the decision. So the neuron is constantly doing this balancing act where it's adding up these inputs in space and time, deciding if the threshold is reached, and then firing an action potential if it is. And here's that image, right? This is actually a closer look at the spatial summation, but I bring this up because you have those dendrites, which are those tree-like branches, you have the cell body, you have the nucleus, and then that kind of, and I'll show it right here. This is the axon hillock. This is the decision point. Okay, this is the all or nothing right here, right? Now, I will say, again, remember, spatial summation is when the signals from multiple synapses at different locations combine at the same time. So in the top row, just below that picture, the top row, you're actually going to see two EPSPs together. Okay, so if synapse A and synapse B, right, A and B, with that gap there in the middle, both send excitatory signals at the same time, their effects add up. Okay, you can see the red line showing how the bump is bigger when both are stimulated. Okay, so you see A, you see B, together, it's even larger. Now, this makes the neuron more likely to reach the threshold. So it's like two friends shouting, jump in at the same exact time. The second row or the middle row are two IPSPs that are together. If you have two inhibitory synapses, C and D, they're active at the same time. Their effects also add up, but it's adding up in the opposite direction. Okay. And that makes the dip in the line even bigger. Okay. When C and D are combined. And it actually makes it even less likely that the neuron will fire. So it's like two friends that are shouting, like, don't do it, right? At the same time, louder together. And then the bottom row is when we have one EPSP or excitatory and one IPSP inhibitory signals that are arriving at the same time. They can actually cancel each other out. Okay, so notice how the red and blue signals balance, right? That line is flattened. Okay, and that's the neuron basically staying neutral because the yes and the no inputs balance each other. So spatial summation is all about the location. It's all about the timing. Multiple inputs happening at once can actually push the neuron closer to firing, further away, or cancel each other out completely. Now let's go to the temporal summation. Um, Again, it's a little different. This one is all about timing. So if you look at the red lines here, these are two EPSP, so the excitatory potential. And you can see that if an excitatory synapse fires once, the bump is small. But if it fires again very quickly, before the first one fades and they bump together, it makes a bigger effect. Okay, so it's like one friend that's telling you, like, go on, do it over and over really, really, really fast, right? The message makes, the repetition actually makes the message more compelling. Now, in the bottom graphs, you'll see two inhibitory potential, so two IPSPs in rapid succession. The same thing happens with inhibition. So if an inhibitory synapse fires repeatedly in quick bursts, those dips add together, which create a stronger break. Okay, so that's like someone that's repeatedly saying stop over and over, stop, don't do it, don't do it again and again until you listen, essentially. So with temporal summation, the neuron isn't necessarily combining signals from different locations. It's just combining signals across time. So just like spatial summation, the combined effect can actually push the neuron either closer to firing, hold it back, or even completely stop it. Now, let's walk through what actually happens during an action potential, right? That's the moment that the neuron fires. So you reach the threshold, okay? Once enough excitatory signals add up, the neuron crosses that threshold and things move really, really fast. So you have the sodium channels that are open, and that sodium rushes in. The first thing that happens is sodium channels are snapping open. Now, sodium, as I mentioned, waits on the outside. It comes rushing to the inside of the cell, and this makes the inside of the cell quickly flip from negative to positive. Okay. You also have the potassium channels that are slowly open. Okay. The potassium begins leaving the cell, trying to really restore some of that balance. So you can think of sodium as the crowd storming in a concert venue all at once, right? While potassium is the slower crowd kind of trickling out. Because potassium is a bit slow to stop, um, the cell overshoots and it may become too negative for a short period of time. And that is called hyperpolarization. It's almost like the neuron is slamming on the brakes after flooring the gas pedal. So that action potential is really just carefully, you could think of it as a carefully kind of choreographed dance of sodium rushing in, potassium flowing out, and then that's what allows the signal to travel down the axon to those buttons or that axon terminal, those axon terminals. And again, the threshold of excitation is about negative 70 millivolts. Now, here's the action potential in full. So this is just a figure, and this graph shows how the electrical charge inside the neuron changes over just a few milliseconds. And we'll break it down step by step. So the neuron starts off at rest, and that flat line is at -70. And we can see that here, -70 millivolts. Think of this as the neuron's, hey, ready but waiting state. I'm ready, but I'm waiting. Now you see the red line, which here is the rising phase. Once that threshold is reached, the sodium channels fly open, sodium starts rushing into the cell. And that's why you see this line shoot upwards so much. The inside of the cell quickly becomes more positive. So it's like a floodgate that's opening. You have the green line, which is repolarization. Okay, so at this peak right here, sodium channels actually start to slam shut and potassium channels open. And potassium flows out of the cell, bringing the charge back down toward the negative again. And the cell is really trying to reset. The blue dip that you see here is going to be hyperpolarization, okay? Because the potassium is a little slow to stop leaving, the cell overshoots. It gets too negative for a very short period of time. And that's why you have this blue dip here. It's like when you swing a door closed and it bounces back a little bit before it actually settles and shuts. And then it returns back to the resting potential of this -70 millivolts. The channels reset, the sodium potassium pump, helps bring the neuron back to its resting level, ready for the next signal. So this whole rise and fall cycle is just one action potential, right? The spike that neurons use to send information, and again, happens in just a few milliseconds. Now, after a neuron fires an action potential, it can't just turn around and immediately fire again. Okay, it actually needs a little bit of a recovery time. And that's where refractory periods come in. Now, there are absolute refractory periods where this is a no exceptions phase. So, right after an action potential, the sodium channels are inactivated. The neuron simply cannot fire again, no matter how strong the signal is. So, you can think of it like trying to flush a toilet right after you've just flushed it, right? The mechanism needs to reset first. And then you have the relative refractory period. So, after this absolute period ends, the neuron is still a little harder to excite than normal. Okay. It can fire again, but it takes a much stronger input to reach that threshold. Now, this is because the cell, as I mentioned in the graph or the image before, it's because the cell is hyperpolarized. It's more negative than usual. And you could compare this to trying to push a swing. Okay. When it gets to the lowest point, a gentle push gets it moving, but if it's pulled way back and you want to reverse it, you're going to need a much stronger shove. So these refractory periods are really what makes sure that an action potential or action potentials only go forward down the axon, and they prevent neurons from firing uncontrollably. So think of it as, basically it's a built-in reset and protection system for your nervous system. So now that we've seen how an action potential starts, let's talk about how it travels down the axon. Now, action potentials move actively down this axon, okay? The process is slower compared to electrical signals in a wire, but it does have a key advantage, and it's non-decremental. That means that the signal doesn't fade as it travels. The spike is just as strong at the end of the axon as it was in the beginning. Now, here's how it works. When sodium rushes in at one section of the axon, some of those sodium ions actually diffuse, and they diffuse onto the next section. And this depolarizes the next patch of membrane, which opens its sodium channels, creating a new action potential. So it's like a row of falling dominoes, right? One falling knocks over the next, and so on and so forth. Now, because these sodium channels are so densely packed along this axon, this creates a continuous wave of depolarization moving forward. That's how the signal keeps regenerating itself all the way down that axon. Now, normally the action potential travels in one direction only, right? From the cell body to the axon terminals, or those buttons. But under special experimental conditions, you can actually stimulate it to go backwards. Okay. And in real life, neurons always transmit forward, right? But you can stimulate it to do that. And that is called the That is called the antidromic conduction. So the conduction isn't about just one big action potential really running down the whole length of the axon. It's really about a chain reaction of new action potentials being triggered one after another, keeping that signal strong. Now let's look at how conduction changes depending on whether the axon is myelinated or not. And if you remember, we talked about myelination. Now, myelin acts like insulin or insulation around a wire. Okay. It speeds up the conduction, it conserves energy. I think I've referred to it before as the highway. Okay. And it's like a fatty insulation that it goes around the axon. Now, instead of sodium channels opening along the entire axon, action potentials only happen at the gaps in the myelin. And if you remember, this is called the nodes of Ranvier. Okay, the symbol is actually, or the signal is actually jumping from node to node. And that is called saltatory conduction. And because the signal really skips over myelinated sections, transmission is faster and requires less energy. Remember that highway. Now, not all axons conduct at the same speed. Okay, larger axons and myelinated axons are the fastest, while smaller, unmyelinated axons are slower. Okay, and this is why reflexes like pulling your hand when you touch a hot stove happen very quickly. Those neurons are heavily myelinated for speed. It's almost like you react before you even actually do anything or think through it. Now, some neurons don't have axons at all. And in those cases, signals spread passively. They get weaker as they go. This is called decremental transmission. It is fine for short distance signaling, but not effective over long distances. So the big picture here is myelin is what makes your nervous system fast and efficient. Without it, the signals are slowing down dramatically, which is exactly what happens in diseases like multiple sclerosis, where the myelin is damaged. So now let's put all of the pieces together and follow a pathway of a signal through a typical multipolar neuron. Okay. So you actually have the postsynaptic potentials that begin at the dendrites and cell body. And they're called postsynaptic because, again, they're receiving from a signal. So this is the after, right? They're coming to the dendrites. This is where the incoming messages arrive. Okay. So right here, all of these little dendrites. Now, it arrives through neurotransmitters that are binding to receptors. And these create tiny electrical changes. And those are either excitatory, gas pedal, right? Or inhibitory, brake pedal. Now, the postsynaptic potentials travel toward the axon. And that's what we are seeing happen right here, right? Where it's with these red arrows, it's traveling down. And it's traveling down to this axon hillock, which is that decision point, right? But they do fade as they go, okay? And this is called decremental conduction, right? It's like ripples in the water that get smaller the farther out they go, the farther out they spread. Now, the summation at the axon hillock is that neuron's decision point, right? If all of those postsynaptic potentials add up to reach that threshold, okay, then boom, an action potential is triggered. Now, that action potential is going to travel down the axon. Once it's fired, that action potential regenerates itself here. Okay, so unlike postsynaptic potentials, it doesn't fade. It's non-decremental. That means that the signal at the end of this axon is just as strong as it was at the start. Finally, when the action potential reaches these axon terminals right here, it triggers the release of the neurotransmitter into the synapse, into the gap before it gets to the next neuron. Okay, and that cycle begins again with that next neuron. So signals start as small graded potentials at the dendrites. They get summed up at the axon hillock, which is the decision point. If it is strong enough at that point, it's converted into a full action potential that travels down the axon to pass the message along. Okay. And then last slide before we wrap up part one. We have been using the Hodgkin-Huxley model to understand how action potentials work. Now, this model was groundbreaking, right? It actually gave us the first real mathematical description of how sodium and potassium ions create these action potentials. But like all models, it's not perfect. The model is a simplification, okay? It works great for explaining some of the basics, but neurons are obviously much more complex in real life. Some neurons fire action potentials even when there's no clear input. So the Hodgkin-Huxley model assumes that input is always needed to trigger activity, but that's not always the case in living brains. And then this model doesn't explain why certain neurons never fire action potentials, right? Even though they still play important roles in neural processing. And in real neurons, action potentials actually can vary in amplitude, in duration, in frequency. And the Hodgkin-Huxley model treats them as very uniform, which is helpful for teaching, but not always accurate. So I always like to give different perspectives on models and theories. Um, every model or theory has gotten us to a point, and this absolutely has. Now, traditionally, we think of dendrites as the input side of the axons as the output side. Okay, so dendrites incoming, axons output. Newer research is showing that dendrites themselves may actually generate action potentials, right? And this goes beyond this original model. So the Hodgkin-Huxley model is still one of the most important contributions to neuroscience. It gives us a foundation, but as science advances, we see the need to update and expand our models to really reflect the complexity of real neurons. So that is it for today. We talked a lot. I will post part two in just a few days. And if you need anything, as always, let me know. Happy to help. Thanks.